Hi, so let's talk about the circulatory system and respiratory systems and a little about gills. These two evolutionary adaptations are the reason that animals aren't all small like nematodes and flatworms. These two adaptations have allowed animals to grow large. Every cell in a multicellular animal has to be able to exchange nutrients and gases in the same way that single-celled organisms do through their membrane just by diffusion. But in a multicellular organism, that outside environment is many cell layers away. So the circulatory system brings the outside to the inside and allows for much more rapid gas exchange. Gills are one of these ways. So gills allow oxygen to diffuse into the circulatory system and allow CO2 to diffuse out into the surrounding water. And lungs serve essentially the same purpose, only now gas exchange is occurring with the air instead of with water. Because for most of the cells in a multicellular organisms, they are too far away to exchange gases with the environment directly, because diffusion in water or in a watery environment is pretty slow. And that's really how these gases are moving around. So gases diffuse from your lungs to your circulatory system and then diffuse from the capillaries into the cells. So all the cells in the body have to be no more than about 2 cell diameters away from the nearest blood vessel for that reason, because diffusion in a liquid, water we're talking about, is slow. Some animals, like the simpler animals, sponges, cnidarians, flatworms, even nematodes, all the cells can exchange oxygen and carbon dioxide directly with the environment because they're only a few cell layers thick, or they have a way, like with jellyfish, the interior of the body is still only a few cell layers thick. So for larger animals, they have to have a way to bring the outside to the inside. So for flatworms, even the reason that flatworms have to be flat and they can't be thick is because they don't have a circulatory system and they don't have gills, and all the cells in a flatworm's body are directly exchanging oxygen and carbon dioxide with the surrounding water. For animals that have circulatory systems, there are open and closed circulatory system. And we'll talk about both of those. So a circulatory system circulates fluid. There's some kind of fluid, which we call our circulatory fluid blood. But for organisms that have an open circulatory system, we don't call it blood. There are interconnecting vessels, and there has to be some kind of a pump, which is the heart, to pump the circulatory fluid around. And the circulatory system then allows much more rapid distribution of oxygen, carbon dioxide to get to the outside, and also getting rid of wastes and getting nutrients to the cells without waiting for diffusion to get all the way there. It brings the source much closer so that diffusion only has to occur across a very short distance from the blood vessel, which would be a capillary in us, from the capillary to the neighboring cells. Insects have an open circulatory system, and so do most mollusks. We call their blood hemolymph because their circulatory system is an open circulatory system. In a closed circulatory system, we have blood that circulates within blood vessels, and now those blood vessels are leaky to fluid. And we'll find out more about that when we get to the immune system chapter. but the capillaries are not impermeable to the fluid that's in our blood. And fluid leaks out, and fluid also leaks back in. So even though we have a closed circulatory system, well, say it's closed, right? Because fluid can still get in and out. Generally, the red blood cells aren't getting in or out. But there are other cells in the immune system that do crawl in and out of the circulatory system. Closed circulatory system, that would be annelids like the earthworms that we dissected, cephalopods, so octopus and squid, and all vertebrates have a closed circulatory system. And remember, we'll say closed circulatory system in air quotes because it is still leaky. So we'll talk a little bit about the organization and the structures in the heart and how the heart works. There are 3 kinds of blood vessels, mostly, and this is according to their size and whether they're carrying blood towards the heart or away from the heart. Arteries and veins are carrying blood either away from or towards the heart, and capillaries are the smallest vessels. And then we have other named types of vessels that are kind of intermediate in size there. And it's the capillaries that are the leaky ones. Arteries are carrying blood away from the heart. Arteries branch into smaller vessels called arterioles, and then finally into capillaries. And capillaries are where the gas exchange and exchange of waste and nutrients is occurring. These networks of capillaries, which are in our muscles and in our organs and our skin and are supplying all of these organs with oxygen and nutrients, these are called capillary beds, and they are also bathed in interstitial fluid and then cells around that, and fluid can leak out of the blood into the interstitial fluid and vice versa. And we'll talk more about that when we get to the immune system chapter. Venules are small veins, and they are returning blood to the heart. And it doesn't matter if the blood is oxygenated or not. Arteries always take blood away from the heart. Veins always bring blood to the heart, whether it's oxygenated or not. Because as you'll see, for the pulmonary circulation, the pulmonary arteries carry deoxygenated blood, but they're called arteries because they're going away from the heart. The pulmonary veins are taking blood from the lungs back to the heart, which is why they're called veins, even though that blood is oxygenated. The chambers of the heart, the chambers that blood enters into are called atria, would be plural. Atrium would be singular. This is Latin for, well, you probably know the word atrium. In a building is the first room that you enter when you enter an office building or something, is the atrium. It's the same root. And the ventricles are the chambers that pump the blood out of the heart. So here we have a human heart, and the top 2 chambers here are the atria and the bottom 2 are the ventricles, and the right side of the heart is pumping blood to the lungs and the left side of the heart is pumping blood to the rest of the body. Fish, including rays and sharks and the bony fishes, have what we call single circulation. So they don't have a pulmonary circulation and a systemic circulation like we do. Instead, the blood is pumped out of the heart and passes through both capillary beds, the gill capillary beds and the body capillary beds, or the systemic capillary beds. And their heart only has 2 chambers, 1 atrium, 1 ventricle, and it's one-way circulation, but when the blood is pumped out of the heart, it goes to the gills where it becomes more oxygenated, and then it continues on to the rest of the tissues, to the capillary beds for the muscles and the skin and all that, and then the deoxygenated blood travels back to the heart. So they only need 2 chambers in their heart because they don't have 2 separate circulatory systems. Double circulation is what we have and what amphibians and reptiles and all the mammals have, and birds. And this means that there's two separate circulatory systems. One circulatory system is pumping blood to the lungs, and the other circulatory system is pumping blood to the rest of the body. So here's what we have for amphibians. Now they have a 3-chambered heart. So blood is coming in, deoxygenated blood from the rest of the body enters the right side of the heart. Blood from the lungs and the skin. Now their pulmonary circuit also includes the skin, because if you remember for amphibians, remember how in our bullfrogs the skin was highly vascularized, and it is part of the gas exchange. It's almost like they have it's almost like their skin is acting like a gill and assisting with gas exchange. So the skin is vascularized by this pulmocutaneous circuit. It returns to the left side of the heart and then is pumped to the rest of the body. Now there is some mixing. They do have a septum in the center of their ventricle, which kind of directs the blood to stay separate, but it does mix. There is some mixing, which means that it's inefficient. So that is that's an inefficiency in their system, because some of the deoxygenated blood is going to mix with some of the oxygenated blood before it gets pumped to the rest of the body. And reptiles have this kind of 3-chambered heart as well. So they do have this ridge in the ventricle that mostly keeps the blood separate. It doesn't completely mix in the ventricle, but it's an inefficient system. So reptiles also have a 3-chambered heart with 2 atria and 1 ventricle, and it is divided a little bit better than the amphibian heart. They actually have an incomplete septum, so a septum is a wall between in the middle of the ventricle, but there's still a hole that allows the blood to mix a little bit, so that's still an inefficiency in the system. Now, if you remember, alligators and crocodiles, they're all crocodilians, they have a complete septum. So they actually have a 4-chambered heart. But it's not quite as efficient as mammals and birds, because there is still some mixing where the arteries exit the heart. So normally for our circulatory system, there's 2 completely separate arteries that leave the left and the right ventricle. But for crocodilians, those 2 arteries are kind of fused where they leave the heart, and so there is still some mixing of oxygenated and deoxygenated blood. So that leaves the question then, what did dinosaurs have? Were all the dinosaurs more like birds, with a completely separated pulmonary and systemic circulation? Or were dinosaurs more like crocodilians, where there was still they have a 4-chambered heart, but there was still some mixing between the oxygenated and the deoxygenated blood? We may never know, because hearts are soft tissue that doesn't really fossilize. So double circulation. So this is what mammals and birds have: complete separation of the pulmonary and the systemic circulation. The right side of the heart pumps to the lungs, and the left side of the heart pumps blood to the rest of the body. So we do have 2 capillary beds, one in the lungs and one in everywhere else in the body. And the arteries are always the vessels that are leaving the heart, even though the pulmonary artery is carrying deoxygenated blood. And so this is a more efficient system, because there's no mixing of the oxygenated and the deoxygenated blood as it leaves the heart. So this allows for higher oxygen levels in the blood. So mammals and birds have a 4-chambered heart, but remember, so do crocodilians have a 4-chambered heart. They just have a little bit of mixing of the left and the right blood. Birds and mammals don't have any mixing of the blood. And so we're all dinosaurs like birds, or we're just the theropod dinosaurs like birds. We don't know. So it's hypothesized that this kind of four-chambered heart with no mixing allows for higher oxygen levels in the periphery because of no mixing, and this is possibly due to endothermy. Well, we do know that crocodilians have they're somewhat endoderms, and so they're endothermic poikilotherms. Crocodilians are, so they do maintain a warmer body temperature, and they do use their energy to maintain a warmer body temperature, but their body temperature does fluctuate enormously with the surrounding water temperature, so poikilotherms. Mammals and birds are not. So perhaps it's this need to have be a homeotherm and have the body temperature very, very little, and that extra oxygen requirement that drove the selection for complete separation of the pulmonary and the systemic circulation. So here's what mammals have and birds, and we have a little person here, but really all mammals have this kind of circulation, not just humans. And if you follow the little numbers here, it shows you where the blood goes. So if we start in the right ventricle with the deoxygenated blood that has just returned from the systemic circulation, that blood is pumped through the pulmonary artery to the lungs. It becomes oxygenated. It returns to the heart, to the left atrium, moves to the left ventricle, and then is pumped through the aorta to the rest of the body through the systemic circulation and the systemic capillary beds. Then the deoxygenated blood returns back to the right side of the heart. This is a little video that kind of summarizes that. I'm not going to show it to you right now, but it's in the PowerPoint so you can watch it. So if we look at the mammalian heart again, the two atria are less muscular. They're thinner, they're smaller. They serve as a return place for the blood, either coming from the lungs or coming from the systemic circulation. The ventricles have to pump the blood harder. They're pumping the blood back out, and the left ventricle has to work much harder than the right. The right ventricle is only pumping blood to the lungs. It's not going very far. So it doesn't need to be pumped under as high a pressure. The left ventricle is pumping blood to the rest of the body. So that is the thickest and most muscular chamber in the mammalian heart. So here you can see the left ventricle is much thicker and more muscular, and that's because it has to pump blood under the highest pressure. The blood is going to travel to the entire body. The right ventricle is only pumping blood to the lungs. Don't worry about the names of the valves here. I am not going to ask you about the names of the valves. You should know, however, the names of the major vessels that go in and out of the heart. So the aorta is coming out of the heart. The vena cava coming into the heart. The superior and the inferior vena cava. The pulmonary arteries and the pulmonary veins. You should know those names and which side of the heart they are either going to or coming from. So the heart contracts in a cycle, and we're not going to go over all the parts of the cycle. You may study that later in physiology, however, if you're going on to take physiology. The pumping phase is called the systolic phase, and the relaxation phase is the diastolic phase. And that's really what we're measuring when we are taking somebody's blood pressure, is we're looking at the pressure in the vessels during these two phases, during the pumping phase and during the resting phase. And that's why these two numbers are the systolic number and the diastolic number. So every time your heart pumps, the pressure increases. So the systolic pressure is the higher number in blood pressure because that's the pressure when that left ventricle contracts. And the diastolic pressure is in between beats when the heart is relaxed. That is the number we are measuring with that lower number. Blood pressure is, of course, something that's maintained by a homeostatic mechanism. There are several mechanisms that control blood pressure. One is vasoconstriction or dilation. If blood vessels are constricted by constricting the smooth muscles in the artery walls, that's going to increase blood pressure. Relaxation of the smooth muscles will decrease blood pressure, and this is the system that is affected by some blood pressure medicines. They cause a relaxation of the blood vessels, which lowers blood pressure. There are other mechanisms involving salt in the blood and salt being removed or retained into the blood. That takes place in the kidney, and we'll talk more about that when we get to the chapter on osmoregulation in kidneys. Gravity affects blood pressure. It will raise your blood pressure if you stand up because you have to keep the blood from draining out of your brain when you stand up. So that's why when they measure your blood pressure, you shouldn't take somebody's blood pressure when they're standing up. You take their blood pressure when they're sitting down and they've been resting for a few minutes to get an accurate reading for comparison. Because we don't want to know the highest we can make their blood pressure go if for medical reasons. Usually we want to know what is their normal blood pressure at rest. So, and a normal blood pressure is about, and a lot of times when we say normal, we mean average. So a healthy average blood pressure would be about 120/70. You can have lower blood pressure than that, and it's perfectly fine and normal. A lot of very young people, like teenagers and kids, have a blood pressure that's as low as 80/60, and that's considered perfectly fine and healthy. But for an average adult over 18, 120/70 is good. We don't want either number to be too high. A really high systolic number, that indicates that you're damaging your blood vessels. If the diastolic number is too high as well, that indicates that the pressure is never getting low enough and that your blood vessels are sustaining damage all the time. And so neither one of those things is good. And high blood pressure, sustained high blood pressure, is very well known to be associated with all sorts of bad outcomes, including heart disease and stroke. So we don't want to have high blood pressure. If you're going into the medical field, you will probably learn how to take blood pressure by hand. Very often it's taken automatically by an automatic cuff, but you'll still have to learn how to do it by hand. So the basic idea is we have these inflated cuffs, which you've all experienced. You pump up the pressure inside the cuff, which it has a little pressure gauge that it reads out what the pressure is underneath that cuff. You want to pump it up to a high enough pressure that the blood is cut off, that there's no more blood traveling to that person's arm. And so generally they pump it up pretty high if they don't know what the person's blood pressure might be, because they don't want to have to take it a second time. If you underguess the high one, then you just have to redo it. Then you need to listen with a stethoscope, and they listen in the crook of your elbow, because what they're listening for that blood to start to squeeze past that cuff as they lower the pressure. So they put the stethoscope in their ears. They're now listening to they're listening to your artery below the cuff. And they should hear nothing when they first put it on because the blood is cut off. And then they turn this little wheel here, and they slowly release the pressure in this cuff. And then they listen. And what the pressure, where they hear the first beat of the blood getting through the artery, that's the high number. That's when your heart is beating, that's the systolic number. What pressure is it finally able to squeeze through? That's your high number. Then they continue to let the pressure down and listen to your heart beating in your arteries. And then finally, when they can't hear it anymore, that means that it's completely loose. And that pressure, then they make a note of that pressure. What is the pressure at which I stopped hearing the heart beating? And that's your low number. So that's how you take blood pressure by hand. So watch, the next time they do it to you, watch what they're doing and watch those steps that they go through when they take it by hand. Because even though we have those automatic cuffs now, sometimes they don't work, or it's broken in whatever exam room you're in and the person has to still take it by hand. So watch next time. Okay, so we'll come back and there's one more video for this chapter.